Hello, everyone. My name is Diana Wall, and I'm the director of the School of Global Environmental Sustainability at Colorado State University and the chairperson of this National Academy Study Committee. In my role as chair of this study and as moderator of this session today, I'd like to welcome everyone here on Zoom. Those of you watching the live stream and all of you who may be watching a little later. The task being undertaken by this committee of the National Academies of Science, Engineering, and Medicine is to explore the linkages between soil health and human health. The committee's full statement of task can be found on the study website, which is available at the link under Project to the right of the embedded video in this meeting. I would like to give some sense of the format of today's meeting. I have a few housekeeping comments to make, and then I will turn things over to Carol Laney, who is the study director with the National Academies, to provide an overview of the study process. Then, following her presentation, I will introduce the speakers for today. We will adjourn the meeting at 5 p.m. Eastern, or whenever our conversation is complete, whichever comes first. To start, I want to note that this is an open, on-the-record session. Interested individuals and members of the press have been invited to join us as observers. This is not a news conference, however, and we will not be entertaining questions from the audience. Reporters who really want to talk with members of the committee are asked to please contact the staff officer, Carol Laney. Also, anybody who wants to submit a written comment or document to the committee is welcome to do so by clicking the provide feedback link on the right side of the study website. You can also subscribe for updates about this study by signing up for the National Academy's Board on Agriculture and Natural Resources listserv, also on the right of the webpage. I would like to remind everyone that this is an information gathering session. That is, that the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone to please be extremely mindful of the fact that the committee has made no conclusions and that it would be a mistake for anyone to leave the meeting today thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or of the academies. In addition, committee members will typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing a draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee, and the committee then must respond to this review with appropriate revisions that will adequately satisfy the Academy's report review committee and the NAS president before it is considered an official Academy's report. The committee is comprised of 15 volunteer experts. I emphasize the word volunteer experts, and their names are listed on your screen. You can find a short biography of each member on the study website. I'd now like to turn things over to Carol Laney, as Senior Program Officer with the Board on Agriculture and Natural Resources at the National Academies and the Study Director for this activity. Carol will give a brief overview of the National Academy's study process.
Thanks, Diana. I just want to tell you briefly about the study process here at the National Academies for anybody who may be joining us today who's not familiar with the institution. The National Academy of Sciences was established by a bill passed in Congress and signed by President Lincoln in 1863. Later, the Academy was granted the authority under an executive order to create the National Academy of Engineering in 1964 and the Institute of Medicine in 1970, which was later renamed the National Academy of Medicine. So today we're the National Academies of Sciences, Engineering, and Medicine, and we consist of the 3 honorific academies, but also the work of scientists across the country outside these academic groups to provide expert advice to the nation on issues involving science, engineering, and medicine. The mission detailed in the Congressional Charter is to investigate, examine, and experiment and report upon any subject of science or art whenever called upon by the government. We fulfill this mission through many convening activities, including consensus studies like the one we're discussing today. Just to share a little bit more detail about consensus studies as opposed to other convening activities we organize, such as workshops and forums, consensus studies are conducted by committees convened specifically for a unique statement of task. The experts who serve on these committees do so as individuals, not as representatives of any organization or institution to which they belong. They also serve as volunteers, as Diana mentioned. Consensus study committees review the state of the scientific literature to write a report that responds to problems identified in their statement of task. That report will hopefully be used to shape sound policies, inform public opinions, and advance the pursuit of the scientific field in question. The study has several stages. So first, a statement of task is defined between the National Academies and the study sponsor. The study sponsor for this particular activity is USDA's National Institute of Food and Agriculture. Next, the National Academies asks for nominations to identify individuals with the necessary expertise to address the statement of task. Each committee should be comprised of individuals with an appropriate range of expertise relevant to the task and also contain a balance of perspectives on the task questions. Nominees are appointed to the committee by the president of the National Academy of Sciences, Dr. Marcia McNutt. And committee membership is provisional until the committee has completed a discussion of conflict of interest, disclosure, and composition at its first meeting. After the committee is appointed, they move into their information gathering phase, which is the phase we're in right now. This is when the committee invites in speakers to present to them in person or via Zoom. Members of the public may also submit information to the committee for the committee to consider. As it gathers information, the committee will also begin deliberating on how to best address its statement of task. They will then begin writing a draft report, and when that draft's complete, it's reviewed, it must be sent out for peer review, and the committee must revise the report as needed in response to the reviewer comments. The National Academy's Report Review Committee determines whether the reviewer comments have been appropriately addressed. And when they determine that is so, they approve the report for release. The final report is delivered to the sponsor and made publicly available. I just want to say a few more words about the committee meeting process. When committees hear from speakers like we're doing today, we call this open session. Anyone from the public may watch the presentations, and if we're meeting in person, may attend the meetings. The committee also meets in closed session to deliberate on the statement of task and devise the best plan to address it. When we meet in closed session, only the committee members and the staff are present. So this is the general timeline for our study on soil health and human health linkages. Today is our 4th public meeting. We have 2 more public meetings scheduled for June and several planned for July. Our next public meeting will be next Thursday, June 22nd. That will be a hybrid meeting with the in-person portion taking place in Washington, D.C. There's more information on the website about that meeting. We'll have another webinar on June 29th about soil contaminants, and information about that meeting is also posted on the website. I anticipate the draft report will be ready to be reviewed in the fall of 2023, and that the final report will be publicly available in the spring of 2024. So here is the first paragraph of the statement of task, and I'll just pause a moment so you can read it. You can also find the study statement of task on the website. And if you go to the next slide, Kat, this is the rest of the statement of task. And as I said, this study is being sponsored by USDA's National Institute of Food and Agriculture. And the study originated with a request from Congress in the last appropriations legislations. Finally, this is our study website. So on the left, you can find the tabs for our upcoming public meetings. The statement of task is under description. You can find the committee members' names and bios, the sponsor information, as well as recordings of the meetings that we've already held. And on the right, in the oval, you can find the link to provide feedback to the committee. And you can also find the form to sign up for emails from the Board on Agriculture and Natural Resources, through which you'll receive notices about our upcoming public meetings. In the event that you need to contact the staff, my email address, as well as that of my colleague Catherine Kane, are there. And with that, I'll turn it back over to you, Diana.
Thank you, Kara. It is now my pleasure to introduce today's first speaker. Harsh Base is a plant biotechnologist at the University of Delaware who focuses on understanding the biological significance of root exudation. His research pursues a multidisciplinary approach of plant biology and chemistry to unravel the underground soil communication process. The output of such studies will not only increase our knowledge about this unique plant defense mechanism, but also assist in the discovery of novel chemical weaponry present in plant pathogens to overcome plant defenses in general. Dr. Bays completed a PhD in plant biotechnology at the University of Mysore in 2002 and conducted postdoctoral studies at Colorado State University. Harsh, thank you so much for joining us today.
Thank you, Dan. Thank you very much for the study section to give us an opportunity to basically talk about our work. And I'm not going to take too much time and just start sharing my screen. And if all of you can see it. So this is a project, and it's very appropriate that what the study section is doing, trying to link soil health to human health, and some of the work that has been done in this regard is also done under the umbrella of this FR grant. So majority of the work that I'm going to show today is not coming out from my lab, but as a group with this particular FR project, which is funded through USDA FR program, trying to link soil health to human health in regards to different agriculture practices, in regards to multiple species, trying to understand how different agriculture practices have led to either a decline or changes or modulation in type of the protein content that we see in grains. So just a disclaimer out that majority of the work that I'm going to show is coming out from this large, very large partnership that we have come across in terms of bringing different type of expertise on board to understand this very, very complex network of what happens in soil health ultimately reflects in terms of our staple crops, which is going into the food chain. So the questions that I'm going to pose today, obviously very appropriate and very much timely in terms of what the study section is doing, trying to find the link in between soil microbes or soil health, ultimately, and its impact on human health. The other aspect, which I'm going to talk very quickly, is how food safety issues are also linked to nutrition and human health, which also kind of comes under the same umbrella of whatever happens below ground in terms of some of the crops, how it changes our perception towards human health in terms of looking into some of these crops. So those are the two questions that I'm going to answer in the stipulated time that's given to me. First, the topic related to the first question in terms of linking the soil health to the human health. And as I said, majority of this work is coming out from this large, very large collaboration of almost 15 different partners. Some of them are land-grant universities, some of them are traditional farms, some of them are actually federal agencies such as USDA ARS bodies. But the majority of this actually stem out of from Rodell Institute, which is one of these largest institute, which basically harbors some of the soil trials that they've been doing where they have not used any of the synthetics or any kind of different fertilizers. So there are like organic practices and different type of conventional farming that they have done over there. So majority of this work actually is coming from Rodell, and we are different partners in terms of trying to find out the different trials linking soil health to human health. So one issue that we've been kind of looking into is the overall global issue of food insecurity, and I mean, this is something which committee is already aware of in terms of how we are trying to kind of engage ourselves in not only increasing the yield to kind of feed those hungry mouths, but also at the same time trying to fortify our crops to deliver that nutritional content that we really want to bring in at the global scale. And this is just kind of this particular slide basically just shows you a snapshot of in preschool children versus non-pregnant women of that age group where there is severe deficiencies. And again, I didn't kind of go into kind of bringing other slides where I can actually show the overall nutritional gap overall globally, which committee is already aware of, in terms of how this is kind of prudent, or rather we have to be on a war footing to basically not only grow more, but also grow nutritious crops. And this particular project that we're working as part of FR grant through USDA is one step closer in terms of understanding when you bring changes in soil, there obviously are going to change, which is going to come across, which will be temporally affected within the plant to see what you're going to see as in terms of grains or the harvest. So one thing which we have kind of observed over the last 60, 70 years of breeding that you've been talking about, and I'm no agronomist, so I'm actually taking this leap of faith to present this particular slide to you in terms of how protein content in grains have declined over time. And this is a snapshot of 1920 till 2002. So think about all the Green Revolution, post-Green Revolution work, and that has basically led to obviously increase in productivity, again, yet not to a point where we can actually still feed all those hungry mouths. But at the same time, these practices, our conventional agriculture practices over the last 80, 90 years have led to this significant decline in protein content. And these are just few examples of some of the staple crops that we are representing here. And majority of them, or rather all of them, are monocots in this case. And that also reflects in some of the essential amino acids, which is reflected in corn in terms of their decline over time. And this is something which we have seen in terms of just one variety, so rather one particular staple crop, which is maize in this case, and has shown that, though we have strived to increase the starch content, but we have at the same time, we are declining, or rather showing our breeding practices have led to decline in protein content in grain, which is also a very, very important gap that we are seeing as part of this FR grant and trying to attempt this particular thing. And one assumption that our large group at FR kind of came out is that the severe decline in protein content is linked to the mineral depletions that you see in different kind of breeding or variety selection practices over time. So Rodell, as I pointed out, is basically, they've been doing this farming system trials for a long, long time, almost initiated in 1981. So they're almost in their fourth decade of trying to understand how different agriculture practices, whether it's conventional till versus no-till, till supplemented with organics, compost, manure, and other kinds of activity that one could do in terms of soil disturbance. And they have done with multiple different species, whether it's legumes, whether it's grasses, whether it's your monocot crop that you can think of. And they have come out with this very, very beautiful system because they do maintain these crop systems and the soil systems, which are untouched as far as tilling goes, as far as any kind of synthetics that you can bring in. So this kind of became a really nice platform for us to test it at multiple scales. Most of the work that we do in the lab is kind of very miniaturized in terms of understanding, say, one gene, one function kind of thing, or one microbe at a time kind of a thing. But when you're talking about such a scale across United States, then it kind of sets up a really nice precedence in terms of testing this, in terms of variation, in terms of species of crop, in terms of different soil system. So this was a really nice idea brought through by Rodell to test this out. And they have done this over a long time. Andrew Smith and at Rodell has shown this, there is if you reduce tillage effects on a long-term basis, you do start to see changes or modulations in yield, but the quality of the grain was not affected at that point of time. And again, this is very species-specific response that people at Rodell have been seeing. So one hypothesis that as a group we came through is that the grain protein content, which was our target, will increase as the availability of soil nitrogen increases over time, whether it's coming through synthetic routes or whether it's coming through natural routes where you're doing organic stuff. And these are some of the results that Rodell actually came through in partnerships with various different farmer groups. And especially in case of corn, tillage was not a significant factor related to the corn grain protein. And again, as I said, this is done at such a large scale that the organic versus conventional management in different locations kind of also led to a lot of variation over time. So results were confounded in both conventional and organic systems in corn, but in case of wheat, they started seeing changes in between conventional practices versus organic amendment that they were doing in their regular system trials, that they started seeing changes in the no-till organics amendment systems. They started seeing some of the regular amino acids or essential amino acids were actually increasing in the grain, especially in case of wheat. So again, you're seeing this variation in between two different crops as far as different field trial goes. So the inference that they were drawing through this particular publication that they came out showing that organic management probably is affecting this protein or overall amino acid content, which may be related to either high soil nitrogen or more biotic activity. So this is kind of giving you a leeway in terms of going from soil health to the human health component. And again, tillage was playing a minor role in regulating wheat protein content, but yet you are seeing these changes, whether it relates to some of the amino acids or some of the protein content that you were seeing in case of wheat, which was completely different in case of corn coming from these field system trials. So one component that which probably brought all of us together in terms of agronomists, ecologists, microbiologists, very basic plant biologists like me, is trying to find a model system. And the one model system which was discovered, or rather found out not very far from us, was at the Hershey and at Penn State Medical School, where they've been working towards this essential amino acids called the argathionine. And this is kind of a very, very interesting molecule, just to say, because it was originally discovered in an ergot fungi in 1909. And then eventually they found that this has also been produced by a lot of fungus and also fungi like bacteria, mainly actinomycetes and actinobacteria and streptomyces and so on. And again, this is something which is not synthesized by plants. Plants don't have an ability to synthesize some of these very essential amino acids. And in the organisms where it's been found to be produced, such as fungi like bacteria, they actually provide the role of antioxidant. And that's interesting in terms of what they were doing. Again, the functional role of these, even in fungi, is not very clear. But humans have a very active transporter to basically take up argathionine. So John Ritchie's lab and Robert Beelman up in Penn State, who are also partner of this FPAR grant, they come up with very interesting hypotheses at that point of time, saying that they started doing this survey analysis where they were looking into overall mortality rate, for example. Again, I'm no expert on this. So Robert Beelman at Penn State, they hypothesized saying that the overall life expectancy, and especially the overall general population, which were up for any kind of neurological disorders, is somehow related to the consumption of argathionine. Again, this is kind of still a leap in terms of kind of saying that argathionine is the silver bullet, but this is kind of one classical example where you can relate that argathionine was shown to be found in patients which basically have less neurological disorders or had less cardiovascular disorders or cardiovascular diseases in terms of this survey that they have done And USA, the overall consumption of argo in USA is so minimal compared to how Europeans consume. Now, we know that argo is produced, or argathionine is produced mainly by fungi or fungi-like bacteria, but plants actually take them up. And it's produced maximum, or in terms of its overall biosynthesis, in mushrooms. So the mushroom consumption also in Bielman's and Ritchie's study showed that was more in Europe compared to other parts of the world where they have seen this obvious disease-like parameters, which is somehow related to argo. So they basically started collaborating as part of this FBAR grant, Bielman and John Ritchie's group from Penn State and overall in this FBAR grant, and they showed this nice correlation. We know that argo is produced by fungi or fungi-like bacteria, but when you start having different kind of ag practices, mainly tilling versus no-till, and within tillage, they start going from tillage, which are basically not that harsh, for a lack of better word, compared to the ones which are really, really harsh in terms of damaging the overall soil structure. They started finding this correlation that when you don't till the soil and you start looking in multiple crop species, in here they did maize, soybean, and oats, they start accumulating more argo in their grain. So this kind of led to that overall continuum that we've been talking about in this study section, in terms of how do you link this continuum of soil health to human health, even if it's related to one particular amino acid in this case. And they found that no-till actually has more argo in grain in multiple crops compared to when you start having disturbances. And this basically led to the overall hypothesis, or kind of a pitch for this particular FR grant, that tillage in long-term systems trials will negatively affect argathionine concentrations in grain, because you're going there and practically and physically disturbing the soil, which basically breaks up all the network. If mycorrhizae or fungus like mycorrhizae, which are very abundant in terrestrial soils and associate with 95% of land plants, basically help plants to take up phosphorus. But again, are argo produced by mycorrhizae or mycorrhizae-like fungi, which usually or historically have evolved to associate with plants. And if you start going through these physical disturbances in soil, you basically create those disconnects in the network, which apparently leads to less uptake of argo in these plants. So that was the hypothesis that as a group we came forward, and then these system trials happen when Rodell on different crops, and you start seeing these kind of, very rarely it happens in science that whatever you hypothesize comes out in glowing gold, but in this case it was kind of shown, at least in case of wheat and corn, that no-till led to more argo accumulation in soil. The one thing we were noticing is that there's an apparent disconnect in between grain and soil argo levels. So the grain argo levels were really high in no-till, but again, the soil argo levels kind of remain constant across the board in both wheat and corn. So we still don't know what's the source of argo at this point of time, and how is it being taken up in plants, because plants lack the transporter that humans have or mammalian systems have to take up argathionine. And so tillage may play a role. That's what's been shown, even if there is a strong disconnect in between soil argo and grain argo, which we can't explain at this point of time. And as a group, we thought that fertility management, meaning how you amend soil, may also play, or rather shed more light in this direction. So the role of AMFs, or mycorrhizal fungi, since the original reports of argathionine production came and showed that some of these fungus produce it, even if it's not very well elucidated in case of fungi. But Wade Heller's group at USDA-RRS, part of this large FR grant, showed that in multiple systems, that when you supplement under controlled conditions, you supplement soil with specific type of mycorrhizae, you can actually increase this uptake or fortify grains with more argathionine. So there is definitely a role of few mycorrhizal fungi that can facilitate the uptake of this overall argathionine in case of plants. And we at our end also started looking at doing this controlled experiment at the University of Delaware as part of this FPAR group. So we were also looking at what happens if we just provide the plants with pure argathionine, because there is always a question mark in terms of where exactly it's coming from in soil. Soil is so complex. Even if Wade's work has shown that if you sterilize soil, you basically cut off that link of uptake of argo. So we started doing more work with spring wheat as a system, trying to see if we add mycorrhizae or pure argo in soil, do we see this uptake? And is it temporal, meaning it's going from root shoot to grain? Is there a continuum of overall uptake or not? And we do see that. We do see over time, under controlled conditions, that argo does go through. Even if you provide pure argo in soil, you can actually see this uptake of argathionine. If you sterilize soil, and even if you're giving pure argo to the plant, you don't see that uptake going through. So there is definitely a biotic involvement in soil, whether it's coming from fungus or fungus-like bacteria. We don't know at this juncture, but we do see this temporal uptake going through, which basically accounts for shoot versus root argathionine, which we have seen with or without mycorrhizae, or pure argathionine. So this is a very neat study, which is coming into fruition in terms of trying to find this connect. And in the same regard, we are also trying to pinpoint where this argo is coming from. Where is argathionine coming from? So we are basically trying to look more detail, trying to use some molecular skills to figure it out. And a student in my lab is basically trying to figure out this whole argathionine biosynthetic pathway in a few of the mycorrhizae as part of the whole group working with Wade Heller's group at USDARS. So she has basically developed this bioinformatics platform to basically look at these EGT proteins and trying to see whether few mycorrhizae or consortium of mycorrhizae do produce this. And it's been shown that some mycorrhizae, especially Rhizophagus class species, have this distinct EGT genes that they can lead to production of argathionine. The issue that we don't know at this juncture, whether it's a consortium of mycorrhizae or a single inoculum of mycorrhizae could do this. And along this regard, we have also kind of looked more into the visualization process, because it's hard to basically grow mycorrhizae with the host. So we are more visual people. So we have developed this nice microscopy technique to basically see if they associate with roots. And if they associate with roots, what's the temporal uptake of argo or not. And in this regard, Alex in the lab is also, because this is a much more clear-cut, or rather an easier system compared to mycorrhizae. So we are also looking at sources other than mycorrhizae, such as streptomyces. These are, bacterial-like fungi, or fungus-like bacteria, you can say, and they are a master of small molecular weight compounds. Half of your antibiotics actually comes from streptomyces species. So, and they have a very well elucidated pathway for argathionine in these. So Alex in the lab is trying to figure it out whether some of the model streptomyces species that we have are producing the same argathionine, and then she's trying to associate this with plant. In this case, we are doing this with wheat to see what kind of transporters are involved. Majority of streptomyces which associate with plants are endophytic in nature, but the ones which we are working with right now colonizes the roots. So we are trying to figure it out if it produces argathionine 1 and if it can go in the plants or not. So trying to kind of club these into going from a very large scale experiment which I don't understand much, compared to trying to do things where we are not modulating a lot of factors. So to conclude the first part, there aren't many conclusions that I'm actually pitching for this particular study section. The idea to connect soil to human health is important. It's very timely, in my opinion, and worth pursuing. And we are probably doing this from a very, very miniature scale and trying to use one amino acid at a time as an example. So the no-till soil data that I showed, part of the large group exercise, definitely shows the microbiome involvement. At this point of time, we are analyzing the microbiome data also in the soil with till, no-till, and conventional, with or without different type of mycorrhizal consortium. So we also want to develop that study further to see if under these different soil agriculture practices, how the microbiome is evolving over time in different species from different conditions. The argathionine source needs to be elucidated further in terms of the overall mechanism that we want to come through as part of the uptake. And then it has to be correlated from different ag practices as part of the overall large grant. Temporal uptake of Argo needs to be also elucidated, in our opinion, basically to see how it goes and just lodges in the grain. And why do you see this disconnect between soil versus grain argathionine content? And finally, the other type of practices, whether it's regenerative or restoration-based ag practices, how does that relate to nutraceutical and fortification? And this study section is kind of very critical and important to basically bring this at the forefront to understand this overall mechanism that we've been talking about. So that's kind of the first part of the study that I wanted to briefly talk about, and I'll just go very quickly to talk about another study which basically we are doing in terms of connecting food safety, health, to nutrition. And this is the work which we've been doing in collaboration with Callie Neal's group at University of Delaware, who's also part of the study section here. So this is something which we've been doing for the last several years, trying to connect how changes in food safety levels could relate to human health and also could relate to different type of nutritional aspects that you're talking about. So you're mainly referring to leafy greens and raw commodities of plants that you eat. And this again relates to some of the opportunist pathogens that you can talk about, mainly foodborne illnesses, which are not very uncommon. 1 out of 6 Americans estimated 48 million illnesses that you can think of. And again, this is all coming from raw commodities such as leafy greens, your lettuce and spinach and so on. If you look into our consumption, 35% of leafy greens are consumed as either salads that we all eat, and these are minimally processed, so to say, and they're all prone to these food-based contamination, which eventually leads to poor nutritional quality and out perception issues in terms of consuming some of these leafy greens. So the relationship in between food safety and plant nutrition, that's what I thought is important to also talk about here in this particular forum. And it's basically the post-harvest processing, which is a very big problem in case of leafy greens, which leads to some of these nutritional issues that you can think of in terms of how important these are in terms of the type of different nutraceutical traits all these leafy greens give out to all of us. Think about alkaloids or isothiocyanates or other kinds of important metabolites that these guys produce. But when plants actually go through this issue, either in pre-harvest or post-harvest, mainly post-harvest, when we are trying to minimize or deter these food contamination, you accidentally or in a way affect the nutritional quality of these particular plants. So pre-harvest and post-harvest contamination in terms of some of these human pathogens that actually uses plants as vectors, and then they come in human food chain, and cause problem for all of us. Whether it comes at pre-harvest, post-harvest, processing issues leads to all these problems. So one thing which we have been doing for the last several years is trying to understand how plant pathogens or human pathogens use plants or hijack plants' defense response to go inside. So this particular movie shows you of a classical plant pathogen, and this is in case of Arabidopsis. And what you see here is a stomates, which are basically breathing pores in plants. They are used in transpiration or gaseous exchange. And in green, you see Pseudomonas syringae, which basically causes disease in tomato and also in Arabidopsis, and also different syringae causes infection in other crops too. And what you see here is these guys actually use stomates as an entry point to cause ingression and cause disease. We saw a similar analogy with human pathogens too. And this is in case of lettuce. And in green, what you see is our human pathogens here. This is Salmonella. And you can see that they enter stomates. And the work that we have been doing for the last 10 years has shown that some of these human pathogens can hijack plants' innate defense response, keep the stomates open, just like plant pathogens, and enter cause disease. The Z stack here on the right shows the bacteria can go inside and cause ingression, and they are present in apoplast. And no matter how much you wash as you do, the bacteria is always there and will cause infection. So one leap which we have taken in this particular study is to try to show that the level of nutrition, the level of food safety, fine, it's an issue in terrestrial, but what about space? How safe are our astronauts and research space stations where plants are experiencing? If you try to grow plants over there, they're experiencing multiple physiological issues as far as changes in abiotic conditions. So one thing which we have asked is, in space, if you try to grow plants under altered gravity conditions, what are the food safety issues lying, and how does that relate to overall this plant health versus human health continuum? So we've been doing this experiment in lettuce and trying to mimic what one would experience in space flight. So these are plants grown in planostats, where we can fool the plant and change gravity lines. And what you see here is plants are actually, they will show a negative gravitropic response of roots. So they're confused. They don't know what's happening. At the same time, we bring in bacteria, in this case, human pathogens, and try to see what happens at the stomatal level or the stomatal defense level. So this is how a plant which is actually experiencing altered gravity shows high ingression rates by Salmonella. And that we have shown in the past, we have shown that Salmonella can hijack plants' defense response. And in space-like conditions where you all change gravity, here we rotate plants at 2 RPM or 4 RPM, that's enough to confuse plants, but not the bacteria. You start seeing stomatal opening, or rather the stomates remain open, which causes more ingression. So, plants are showing this physiological stress by keeping stomates open, but one particular opportunist is trying to use that trait and trying to ingress. So this is an ongoing work. There are some conclusions in this particular area that we've been doing. We know that post-harvesting processing to detect or deter food contamination may lead to poor nutrition in leafy greens by the amount of processing they go through. We show that there was a link in between human pathogens and plant pathogens in a way how they infect plants. We show that altered gravity. So yes, there was a change here in the terrestrial space. There was a change here in this International Space Station as far as altered gravity goes, and how plants will tackle pathogens at that point of time. So key signaling components that we are working at the stomatal level to figure this out, both under no rotation or altered gravity conditions. And then again, as a big pitch, and again, this is something which is very raw at this point of time, to figure out what innovative technologies which involves breeding could come into this particular space to deter or kind of abate this human pathogen response that you were seeing in all the routine recalls that you see across. So a lot of people have worked in this 2 projects in the lab, and I've just highlighted the 2 projects that I talked about. Huge contribution coming from the overall partners that as part of FR grant. And majority of stomatal work was a collaboration, a very fruitful collaboration between our lab and Callie's lab. So with that, I'll stop, and I'd be happy to take questions. Thank you.
Thanks very much, Harsh. That was really interesting, the 2 different kind of sections that you pulled out for us to consider. We've got time for about 1 to 2 questions. If there's any questions, please raise your hand. And I should say the little hand thing is down at the bottom under reactions, I think. Hi, Callie. Okay, there's going to be, oh, there's a hand. Welcome. Go ahead.
Okay, I'm sorry. I have a question about the tillage. Thanks, Harsh. That was a great presentation. When thinking about the no-till of the soils, is there a time length that seems to be important there, like several seasons, or or is there any thought to that type of a situation, a longevity issue?
I think that's a great point. I think majority of work that's what I showed as part of the published work that came out from Rodell as part of this grant is kind of one season. It's basically one season, but I think it's a great point. If no-tilling as a part of practice could be continued across multiple seasons, then yes, you at least I can speculate here if I dare to, that it could be definitely bringing a lot of changes, and this will come out from the metagenomic study that we are doing at this point of time. So even from one season, if there was a change across multiple geographical locations within the United States, then we could come across. And again, this is species-specific response that we're seeing. What works in corn doesn't work in wheat. What works here doesn't work in Montana. So this is something which is very, very, well, it's speculated and it's understood because you do see different soil and different type of climatic conditions. But to answer your point, I don't have an answer to your question, but I think expanding beyond one season would be a great thought. And I can definitely convey this to Rodell because, you know, they are the masters of doing this large field trial. I can't even do a greenhouse experiment. So.
Okay, we have one more question up here. I'm not sure I'm reading the hand right.
Oh, is it me?
Yeah.
Okay, great. Thank you very much for that presentation. I'm just curious, you said that there was some relationship between soil nitrogen availability and protein content in grain, and you had some relationship maybe between mycorrhizal fungi and this compound that I don't even know how to pronounce. As a committee, we're obviously thinking about what specific things we can focus on in terms of crops to have that linkage between soil health and then crop and human health. So in addition to grain proteins and that other compound, what are you focusing on? What do you think are important?
So I think it's we always think growing plants is an isolated effect. We never think about growing plants as more of an extended effect, meaning plants are not growing in isolation. So they have these extended holistic genome that you can talk about, which is basically microbiome. And so we are moving even with one particular amino acid, which at this juncture, we don't know where it's coming from. Whether it's coming from fungi, whether it's coming from consortium of fungus, whether it's coming from, actinomycetes. So I think apart from enriching plants with, say, for example, this particular amino acid, I think the overall pitch of focus, I can't say focus because, but the overall thinking should go in the direction of how this is all a continuum. Meaning, if you disturb one thing based on one ag practices or ag practice, how that continuum breaks. So it could be protein content, it could be vitamins. So there would be more examples that could come out from a study like ours, say, for example. So where people can look into other components, say we grow a lot of lettuce, spinach, broccolis. They have huge impact in human health, for example. Some of these isothiocyanates they produce are anti-cancerous. And it's been shown at clinical trial levels, but we don't know how ag practices are affecting that. Or buying sachets of broccoli.
Enormous list of variables.
Yeah, so this is just one, I would not even call it a silver bullet. It's just one example, and that's also not been shown, very, very nicely at this juncture that indeed this is a continuum. But yeah, as a study section, you can talk about, plants are the best chemists, so they make wonderful things.
Thank you.
Thank you very much, Harsh. I think we're going to move on now to our next speaker, which is Jude Mall from USDA. He's a research ecologist with the Agricultural Research Service of the USDA, and his research program determines how land use management and agroecosystem decision-making will impact soil health and plant and microbial ecology. He studies the microbiomes important for sustainable ag production by understanding soil ecology, biogeochemistry, and plant genomics. These results are used to build a sustainable, productive, multifunctional agroecosystem. Biochemical and molecular ecological methods are used to identify the linkages between the landscape scale management decisions and the micro scale, which is quite a spatial scale to look at, and the microbial and plant molecular processes. He completed his PhD in horticulture and biogeochemistry at Cornell University. Welcome. Thanks so much for being here.
Thank you very much, Diana. And I want to thank the committee for giving me the opportunity to speak with everyone today. Can you see my slides okay?
Yes.
Okay. Great. Well, I'm going to present kind of a different talk to accommodate all those scales that Diana was talking about. I'll speak in very general terms and give the committee an overview of some of the challenges and progress that we've made in understanding how farming systems management impacts greenhouse gas production. And at the end of the talk, I'll make linkages between those greenhouse gases produced from agricultural systems and threats to human respiratory health. And those are projects that are ongoing right now. So I'll just give a brief overview of what I'm going to tell you about today. I'll show you a typical farming system, soils from a typical farming system, and some of the spatial considerations that we need to consider when looking at production of greenhouse gases. I'll discuss some experimental and modeling approaches to understanding greenhouse gas production in ag systems. And then I'll describe some links between the microscale soil processes and landscape level phenomena that ultimately has the potential to impact human respiratory health. So this picture we're looking at is the Farming Systems Project, and it's a long-term agricultural research site in Beltsville, Maryland. It's one of the legacy sites for the USDA LTAR network. And we've been studying cropping systems here for about 26 years. And there's a complementary set of systems that we look at, including no-till, conventional till, as well as organic grain production systems. And these are representative of the mid-Atlantic systems, which generally produce corn, soybean, wheat, and some alfalfa. So we study a wide range of agronomy and plant physiology, production level questions. And we use this study site as a test bed to study all different types of issues that may occur in agricultural systems, including measurement of greenhouse gases. So the U.S. Agricultural sector is the largest controllable source of ammonia and nitrous oxide. So that means management decisions that we make result in increased or decreased production of these greenhouse gases. And these gases contribute to what we call atmospheric NOx. So these would be nitrogen and oxygen containing compounds, and they may have a number of different chemical formulations. They cause greenhouse gas radiative warming, and as well as contribute to degraded air quality. Some ammonia and other greenhouse gases are precursors to particulate matter size classes that are of issue to for human respiratory issues, and some papers have suggested that the indirect effect of this may be the premature death of up to 17,000 people a year. And I put my references in here just because I know I'm not a human doctor, but this paper was produced by the National Academies as well. So I hope that everyone goes and looks at those to gain further information. So greenhouse gases, although they may be a problem at the global scale, they're produced by microbes. I always have fun reminding my colleagues of that we talk about greenhouse gases and global models and we still have lots of questions about the microbial processes that control and or regulate greenhouse gas production. So we must zoom in quite a bit. So we're going to zoom in, and there now we can see these soybean rows, and what this points out is sort of the beginning of this heterogeneity in the field. Soybeans are planted all in rows, and that's for convenience of planting and harvest and management. If we zoom in even more, okay, now we're kneeling down in between the rows of soybean, and I've dug a hole here. If you can see right here is a little hole, and we're looking at the soil, and what we're looking at is a typical sassafras series soil, and that's actually the state soil of Maryland. Very interesting. And what is described here to the left, or my left of the screen, is the way we talk about soils and the different types of classes of soils. And the reason we do that is because they have different biological, physical, and chemical characteristics and attributes. And so we see these two different colors here in this soil pit. And one is our AP horizon, and one is a BA horizon. So let's look at that a little closer. So now the soybean rows we're kind of looking right down the row. And now we're looking into this soil pit, and we see coloration differences among the different horizons. This blue line represents basically the soil surface. You can kind of see where we have a pit that's down the row. And now I've shown you where the AP and BA horizon are. If we were to look at the chemical descriptions of this, we'd know that geomorphically, the AP horizon is younger. The reason why it looks like this is because this field had been plowed. And there was some comments earlier about how long the impacts of plowing last in a field. Well, this field was plowed about 50 years ago and has not been plowed much since. And that change in color is the result of that plowing that occurred back then. So you could imagine the destruction that occurs every time we plow. It's very difficult to find an agricultural site that's never been plowed, except for prairie lands or rangelands. So in our description of the soils, we'll also notice that this BA horizon is more acidic. And so those are just two aspects of the heterogeneity that exists within the soil horizon. Okay, so greenhouse gases are emitted from these soils through the processes of biological activity. But as you might imagine, the processes that are occurring in this BA horizon could be quite different than the processes that are occurring in the AP horizon. And we actually know that this is the case, and it points out some of the difficulties of trying to understand the total emissions from a agricultural field, whether it's a particular type of practice or a particular type of crop that we're looking at. As we look at on the left-hand side of the screen, by depth, we have heterogeneity through soil forming factors such as tillage, roots growing through the soil, bioturbation from worms and other soil biological organisms, hydrologic flows affect the heterogeneity at depth, as well as gravity or sedimentation. During these bioturbation events, we get sedimentation as well. So that's going vertically in this landscape, and then horizontally, we have landscape and field heterogeneity that result in productivity hotspots, and this is from residual roots, low topography in the field, or within the soil, restrictive layers such as compact zones or wheel tracks going vertically along the fields. And they may be alternating with planting rows. So you can imagine all of these physical factors are impacting the environment in which these microbes and other organisms are living. And that in turn is impacting the production rates of greenhouse gases. To add more complexity to the mix, farmers can add fertilizer to the field in different forms. And I have two examples here. So there's yellow dots along the soil surface. That would be when a farmer goes out and just spreads a fertilizer, maybe in pellets or something like that. And that's a typical way of fertilizing a field, and it's just broadcast evenly across the field. Another approach is to use a subsurface band of fertilizer in which the soil is cut open and the fertilizer deposited below ground. And the idea is that this fertilizer diffuses out over time to these roots. And the fact that the ammonia and or nitrate is below ground and not exposed to oxygen results in conservation and reduced ammonia emissions from fertilization. So it's there's a sense of conservation there, and your subsurface banded fertilizer is less likely to wash away if you were to have a rain event. So at the landscape level, geologic history and the environment shape soil development. At the field scale, spatial patterns can take decades to develop, and then they can be destroyed rather quickly. So we have a very dynamic landscape in which we're trying to understand these types of emissions. And farming systems management impacts soil biogeochemistry to varying depths. And that is partly to do with the parent material, the geology of the area, and partly to do with the types of farming system. So in some cases, it's only 30 centimeters deep, and there are some soils in this country, for example, the Palouse region out in the west, where 2 meters of soil may be impacted by our surface level management decisions. Okay, so as scientists, we're persistent, and despite this heterogeneity that we see in the field, we think, we know that greenhouse gas production, it only has the few components necessary for this to occur. We need the organic matter and fertilizer, which is really the substrate. Temperature has to be right, enough water, and the pH is in biological reasonable zone. And with those components and the microbes that can perform the process, we'll get greenhouse gas emissions. Now, these variables, you think, oh, well, we can model those. That's great. And we've been doing that for many, many years. So this is a very common greenhouse gas model called the DNDC model, and it's a process-based model. At the top, we see some very broad ecological drivers that we know impact the overall state of the soil. And then we have some middle level soil biophysical types of attributes, which have to do with the arrangement of soil structure and the ability for gases and water to move in and out of the soils. Down in this yellow section down here, these are all microbial processes. And these three processes in particular, there's room for improvement. And part of the reason is, as these models were developed in the 80s, computers were much slower. And so parameterizing these models usually resulted in finding a kind of average kinetic rate, or KM, for these processes and running the models with these static KMs, or kinetic rates. And we know that in nature, biology does not behave that way. And in part, that's a limitation of our compute power and our ability to use multiple threaded computers to do complex calculations and modeling. So part of our goal in our ag systems lab, in which we, as I mentioned, study everything from the agronomy down to the greenhouse gases, part of our goal is to work with modelers and identify areas where there's need for improvement in the models. And then as experimentalists, we go out and find or generate the data for modelers to put back in those models. So we've been working for a long time on these modules to try to better understand how these soil climate and vegetation factors impact those kinetic rates. And just the act of putting a dynamic rate in the denitrification module has been shown to improve model predictions. So we have some evidence that there's plenty of room for improvement here. And I'll just point out that in 2022, we gave a series of seminars at the Tri-Societies, the Soil Science Society of America, Agronomy Society of America, pointing this exact issue out. And we gathered together lots of modelers, microbial ecologists, and we talked about how we can go about improving these models. And that's happening in many different research groups around the country right now. Okay, so despite this heterogeneity that we see in the field, we think, well, if we know quite a bit about environmental sampling, and if we sample the right way, we should be able to control for those microscale heterogeneous zones, so to speak, and work towards a sort of an average denitrification or greenhouse gas emissions rate from this strip. So we've done this for many years doing this type of sampling. You get a sort of an average of what that soybean plot is doing. And we thought if we looked at greenhouse gas emissions over the course of a year and measured the microbes and looked specifically at the abundance of the genes that are needed for denitrification, we could get a real good idea of how the microbial community changes and then subsequently results in more or less greenhouse gas emissions. So this is a representation of the abundance of a variety of genes needed for denitrification. So vertically, we have the 16S gene, which is a general bacterial marker, near K and near S, which are both genes that are in upstream portion of the denitrification pathway and result in the release of nitrous oxide. And then we have NOS-Z, which is the very last gene in the denitrification pathway, and takes nitrous oxide and converts it to harmless N2. And so what we did here was we looked at conventional and no-till system over the course of a growing season. And on the y-axis, we're looking at the abundance of these genes in each of those columns. The dashed line is the soybean crop, and the solid line is a wheat crop. And these are grown side by side. So environmental conditions are exactly the same. And what we see is that we can detect 1.5-fold difference between the abundance of these genes at different times of the year. And this is very important to know from the standpoint of how microbial populations, just the population size, can change over the course of a year. And really, this is just the microbial community growing and shrinking based on availability of root exudates, water, and other soil components that are necessary for growth. Excuse me. So if we look at these results and we look at the subsequent gas emissions, and so on the y-axis here, we have N2O flux, and on the x-axis is time, and I've put in some indications of different types of field events. You see these peaks of N2O occurring at different times of the season. And when we looked at the abundance of denitrification genes and the presence of a spike in greenhouse gas emissions, we don't see a great correlation. And in part, this may have to do with the regulation of these genes at the microbe scale, and the presence or absence of the genes may not be as good a predictor of activity of greenhouse gas production processes as measurement of the mRNA or enzymes themselves. But what we did learn from this study is that the denitrifier populations vary among these different farming practices. And that is in this paper, and it shows that the farming systems type, meaning no-till, conventional till, or organic, results in significantly different populations of denitrifiers, and that the crop species in the rotation impacts the population size of denitrifiers by over an order of magnitude, depending on the physiology or the physiological state of that plant. Okay. So if we go back to looking at this DNDC model, and we think, well, taking samples throughout a whole entire field and trying to mix them all together and get an average is not a fine enough resolution to better predict greenhouse gas emissions. Yet we know that if at a small scale, if we parameterize this model with information like population size of denitrifiers, we do get better predictions. And when we were thinking about this, we thought, well, the answer is right in front of us. Really, we need to run a model. We need to run a model here in this AP zone, and we need to run a model in the BA zone. Maybe it's a smaller impact on that model because not as much gases are coming out. And matter of fact, we probably need to run a model over here for this zone where these roots are, and on this zone. And so you're starting to get a picture of how the heterogeneity at this scale can impact our ability to apply models with parameters that we can physically measure. And so we wanted to design an experiment in which we could do this. And so what we did was, this was an experiment designed by Stephen Mirsky, Hannah Parfenbarger, myself, and others. And what was being studied here was one of these subsurface fertilizer bands, and the distribution of nitrogen from this fertilizer to the planted plants. And what we're looking at now is just like that soil pit. We have two rows, in this case, they're corn plants, and then right in the middle is a fertilizer band. And we sampled at the root of the plant, halfway to the center, and so forth, all the way to the fertilizer band. So the way this is reported is 0 is the center of the row, and 38 is each row on each side. And what we wanted to do was these lines here, these are soil samples. We took probes and pushed them into the ground to 30 centimeters, and then cut up the soil core into these 5 centimeter fractions. And then so this is what the sampling would look like. There would be these samples across here would be in these rows, going from one plant across the row to the other. And we could put greenhouse gas measurement equipment on top of the soil and measure over this fertilizer band and adjacent to it, and look at our greenhouse gas measurements to determine if putting this fertilizer band in was helpful at all in terms of reducing overall greenhouse gas production. We were very interested in applying our what we learned from our previous study in terms of the dynamics of the microbial populations to this study, to understand how placement of this fertilizer band and or growth of plant roots can interact with the microbial community in the context of denitrifying organisms. And so what we're looking at here is the quantitative assessment of the 16S gene copy number to depth. And the reason it's represented like this is because of resource limitation. We pooled the samples going out from 0. So sample 5 centimeters from the band was pooled. The 10 centimeter samples were pooled. And we processed samples for just 1/2 of it. If this is what it would look like if that was mirrored over, showing the hole between both plants across the whole row. And you can see the fertilizer band in the center, and you see so you see increased 16S or bacterial abundance around the fertilizer band and around the roots as well. But I'm going to show the rest of the data just in this 1/2 so you can get an idea. So we looked at ITS, which would be a marker for fungi, the denitrification genes near S and near K, as well as NOS-Z1. And the general take-home message here is that we see a sharp decline in gene abundance in depth. We've done follow-up studies to confirm this, and it's a very depth in soils is a very strong effect on total microbial population abundance. But what we also see is variation in the distribution of certain denitrification genes and not others. So even in this particular soil, this near K, we call it a haplotype or a genotype, is dominant compared to this near S, even though these are both homologs in this case. We see some spatial heterogeneity laterally from the impact of the fertilizer band, also the impact of the roots themselves. So from our results, we found that greenhouse gas production in these systems were really a combination of multiple different functional types of organisms. Where the fertilizer band was, and this is poultry litter, which has lots of ammonia in it, nitrifiers, N2 reducers could dominate, whereas where that nitrogen source was interacting with the carbon produced by the exudation of the roots. We saw denitrifiers dominating in the plant roots. So this again shows that by designing the experiments in particular ways with an understanding of the heterogeneity that may exist in the soil, and some of this may be induced by our farming system practices, such as planting rows and fertilizer placement, that we can start to get a better understanding of who the players are that are producing these greenhouse gases. And this scale is important because in some senses, this looking between the rows about 30 centimeters deep is really like it's a manageable unit of farming. This is going to be replicated over and over and over again throughout the field. So if we can understand how a slice of the field operates and which organisms are active in response to our management decisions, then we can take that type of information and upscale and start to say, okay, well, it's happening in this square meter, then we can go on and project to the rest of the field. So greenhouse gas models are good at capturing coarse fluxes in greenhouse gas, but need improvement among, especially among diverse cropping systems where we have different crops and different practices. So the model modules that represent microbial processes are mostly static in terms of their parameterization, whereas we know soil biology is diverse and heterogeneous. Gene abundance of the greenhouse gas producing organisms is not enough by itself to predict emissions, but our modern molecular techniques can capture some of that population dynamic and tell us about different players who may have different roles in greenhouse gas production. And the environmental and genetic data suggests that multiple greenhouse producing processes occur in the soil column concurrently. So we have multiple processes occurring and those interact. And that knowledge can help us in the future trying to take these complex environments and scale them up for modeling. To that point, ultimately, we want to model like greenhouse gas production using remote sensing. And that will allow us to scale to the scale we need to in order to make predictions for, say, the US production of greenhouse gases and the total loss of nitrogen fertilizer to denitrification and or gaseous loss. So soil temperature, water, and gas emissions can be measured from space. That's great. That allows us to be able to at least get part of the way there in terms of modeling at the landscape scale. So we thought of these ideas and we were thinking in some cases when we're not sure what mechanisms may be at play, sometimes we just look to two systems that have extreme differences. And we wanted to take that approach to see if we could try to upscale from soil scale processes to space detection of the gases. So if we upscale from here, and then that's what that plot looks like from an airplane, and all the way to the country. If you get to this scale, then you have to collaborate with an atmospheric chemist. No, not really, but in order to enter this sort of world of science, I've started collaborations with Anne-Marie Carlton, who's at the University of California, Irvine. She's an atmospheric chemist who has been working for a long time on the impacts of human activity and the resultant air quality and or composition. And she does a fair bit of modeling as well. And so we got together and started talking about greenhouse gases, their impact on large regions of the country. And in particular, Anne-Marie was interested in this particulate matter 2.5 measurement, which is I believe, an EPA regulated class of air pollutant. And as I mentioned earlier, ammonia and some other greenhouse gases act to or are considered precursors to some of these compounds. And what Anne-Marie was noticing was that in some of these PM 2.5 locations correlated quite well with agricultural production zones, and in particular started pursuing the idea that some of these detected hotspots may be around animal production units. And so this Anne-Marie made this chart here with some earlier NASA data. You can kind of see the resemblance of the United States, and this is the NH or the ammonia mixing ratios, and the circles you see there are the animal units. So we see some correlation, but there are clearly areas that are producing a fair amount of ammonia that are not dense animal production zones. So Anne-Marie and I wanted to explore this further, and in particular, we were interested in a new NASA mission, and it's this ARIS instrument, which can detect ammonia mixing ratios at a much higher time scale. So this satellite goes around the Earth and allows us to get measurements of hot spots and hot moments of ammonia production on about a monthly time step. These instruments are improving by the year, and once we started to see this type of data, we thought now we're measuring greenhouse gases at enough of a dynamic rate that we may be able to take a model like the DNDC model and apply it to one of these hotspots. And so that would require getting the temperature, water, soil type information. And what we wanted to do was go in and see if our measurement of microbial populations and or enzymatic activity would play into the prediction for greenhouse gas production in these hotspots. So what we have here, I should have said this earlier, but March, April, May, and then June, July, August, September, October, November, December, January, February. And we see that hotspots appear and disappear at a seasonal basis. And these appear to be related to animal production schedules and fertilization schedules around the country. So we see a connection between the timing of agricultural practices and these hotspots, which is further evidence that they are ag-related. So seasonal remote detection of greenhouse gas emissions coupled with additional USDA and NASA provided data layers, it presents a possibility of testing these prediction models at regional scales. And part of the reason we want to do that is because it's unclear if the observed hotspots are a result of emissions across the whole region, meaning areas of high emissions may be a county or a watershed as large as that. And that could be the case if the environment is saturated with nitrogen. And that has been suggested, and there's some papers below here that suggest that we've entered this time where because we have a agricultural matrix sprinkled throughout our nation, that we have saturated our environment with nitrogen. And in some of these locations, you may have higher denitrification rates just in general. The other possibility is that these hotspots are really a function of super emitter sites. So a particular field that manure and or fertilizer has been dumped on at extremely high rates, maybe a feedlot or someplace where you have very highly concentrated sources and processing of nitrogen, manure, something along those lines. That's not known. This could be an example of a very concentrated hotspot that is emitting greenhouse gases, and through mixing in the lower atmosphere, the satellites can detect this at county scale. So what we did was we wrote a grant to NASA to use their satellite instruments to go in and test this. And this is one particular location, Y Mills, Maryland. And our approach was to find an area that's in a hotspot, use soil maps to match soil types and slope and those other factors that we know play a role in biogeochemical processes, and then identify agricultural and non-agricultural zones within that hotspot and test each of these zones for denitrification rates, activity of denitrifiers, basically all of the parameters that we would predict go into greenhouse gas emissions. So we have 4 locations identified with ag and non-ag locations in each of them. And these are in 4 hotspots around the country that we see are seasonally episodic, and we think are linked to agricultural production. And this is a pending grant where our plan would be to go in and conduct these measurements and input those parameters into our into dynamic modules for those denitrification models. So paired ag and non-ag sites could be used to determine if land use type is the primary source of greenhouse gas production, or if the landscape itself is saturated with reactive nitrogen species. And this is something we don't know about our country. As we accumulate and process more data, we are able to develop multi-models which allow us to link differentially active zones in the soil. So that idea that the AP horizon might be functioning different than the BA horizon, that's already being taken into consideration in some models, but our computing resources now allow us to iterate these models hundreds of thousands of times. And that is one approach that we're taking to try to capture the in-field heterogeneity, with the thought that we may have a suite of models that describe a management unit within a field, and then we can multiply that out to whatever space that type of farming system covers. This information can allow more accurate estimations of the impact of land use distributions on the landscape and potential to predict landscape hotspots and hot moments in space and time. And ultimately, continual estimations of greenhouse gas emissions may allow for better assessment of regional assessment of high respiratory health risks, or something like low air quality periods of time. And so that would be maybe something along the lines of a air quality rating, where we know that ammonia and other NOx gases are very high at this time of year in this particular region. So our proposal is still pending, and I just want to thank some of my colleagues who have helped out with this. And I have, at the very end of this, since I didn't present a lot of data, I have the references for lots of this work. So with that, thank you very much, and I'd love to take any questions.
Okay, thank you very much for this scaling exercise in our brain too. I think we have time for a couple of questions. Well, one or two questions for you, and then we better open it up because we only have a few questions after that. So if there's some committee members that have questions, please raise your hand. And I see Kelly has her hand up. I think that was right.
Yeah, that was right. That was right. Hi, Jude. I really enjoyed your talk. And so I've been thinking a lot about, or we're talking a lot in our committee about microbial diagnostics or indices that we can use. And I saw that you were using DNA as an indicator, and maybe found that it wasn't necessarily predictive, but it was informative still. And I appreciated you discerning the two. Do you have any data to suggest that, like, if we, I mean, in this world, if we could imagine a world where we could really just get to RNA and it was easy, do you think it's a technology, or do you think it's actually the microbes aren't that, like, so if you could do RNA or you could have some activity measure, do you think that it would provide a better signal, or do you think it's just like, hey, that's not a place for continued development in terms of technology? Like, do you have any instances of any RNA data maybe that suggests that maybe
yes,
your seeding doesn't change, but your activity would be more responsive and therefore more?
Yeah, we've seen that mostly in controlled experiments. You know, I think in some ways, if we identify the basic rules of life for these microbes, we don't need to look at RNA, because ultimately microbes are responsive to their environment. Mm-hmm. and if we know the conditions exist that would result in greenhouse gas production, we should be able to predict that.
That ties into my second question, which was when you went into remote sensing, and then I will turn over the floor. I promise, team, I won't hog anymore. So then you went into remote sensing, and you were like, can we predict essentially microbial functionality or infer microbial functionality from like high-res data that we can get?
Yeah.
And so, is that where you were going with these rules of life? Like, if we knew water, we knew soil texture, and we knew these formulas, then maybe we can get around microbial activity and we can approximate it. And what is your thinking? I mean, some sites that works, but we have some sites, I know, in wetlands and other sites where we find that that's not enough, but we don't have we don't have the variables, right? So it sounds like from your intuition, it's like, okay, we might need more metrics to kind of confine that dynamic, but it doesn't have to be microbial. Yeah. Would that be a...
Well, so for the remote sensing part, I'm very much of the mind that we have to step back and pick kind of the easiest thing to look at. So where those maps, the reason I produced those maps was so we could match soil type and not be in a wetland, essentially put everything on the same scale and just ask the question, under this hotspot, because from the satellite, we can't tell if it's this farm or that farm. It's at almost a count or less than county level. But
okay, I didn't realize the resolution you had. Okay.
Yeah, part of the goal would be two adjacent land use types that are under a hotspot or within a hotspot. Are they both emitting at the same rate? Is it regionally, is that whole entire area hot?
Got it.
Or is it just a pinpoint that is like a smokestack of greenhouse gases? And the satellite just picks it up because it spreads over that natural area and over that wetland. And so we won't be able to tell the difference. And the part of the reason we want to do that is if we were to identify that these were just super emitter sites, then in our estimations of the total land area that's producing greenhouse gas, we can say, oh no, that forest is not going to be a contributor. And it's only the land area that is in, say, feedlots or chicken production systems. So it's really that second part was really just to
Sorry, I was talking about the second part of the first part. Sorry. So
Yeah.
the little greenhouse gas where you tried to estimate from, not the larger second part.
Oh,
but
okay.
Yeah. So, because I was trying to get at this rules of life translation. I mean, I think it's an interesting idea, but I don't know if I only work in wetlands with greenhouse gases and estimating, and it doesn't work there. And so now we're like, maybe we need more biological information. Maybe,
Yeah.
so we're trying to figure the same kind of question out. So I like the idea that, like, what are the responsive variables we need to inform greenhouse gases? So I thank you for the thoughts. I'll go through your papers. I'm super excited.
Yeah, I hope I wasn't more confusing in my answer.
No, you weren't. I'll turn over the floor to my colleagues.
Okay, and can we invite Harsh back? And so if we have questions, does any of the committee have some questions for both speakers or one or the other? I know Harsh is still around. There he is. Hi. I'm hanging out here because I know somebody will come up with a question right when we have to stop. And so, yeah, I found these to be, oh, hi, Lindsay, go ahead.
I enjoyed both of your talks, and I actually kind of have a question for both, two separate questions for both of you, if we have a little bit of time. If I could start with Harsh, just to go back to it, because you were the earlier presenter. I was wondering if you've looked into if there's an interplay specifically between legumes and the amino acid that you were talking about, whether nitrogen fixation from legumes and the added protein that usually comes into those crops as a result of that has any type of influence on the levels of those amino acids. I know when you were showing us the data in terms of like those levels in like mushrooms and fermented products, if there was any difference between like the non-legume grains and the legumes as well. Does that make sense?
Yeah. I think I don't think it's been done that widely across multiple species. I think the data that Rodell shared with me for this particular talk was they have done a lot of analysis on mainly grasses. So looking at the staple crops, oats, wheats, and corn, and so on. And that's been done across multiple places. And all of them, wheat was one of the ones which actually came out more reproducible in terms of accumulating more argo. And then from Wade Haller's trial from USDA, he could show that in wheat and oat both. Though field data of oat was kind of a little bit confusing, we still can't find the soil argo versus grain argo connect, meaning across multiple ag management practices, we are not seeing that connect. But to your question, legume was not significant, at least from one trial experiment. And that could also be related to, how conventionally we grow legumes. So there is so much of residual nitrogen, which is over there, and that basically drives all the native populations of at least Cynorhizobium. So I think that's very interesting. For some reason, and only because the field system trial results showed wheat to be more reproducible, we at our end, at a very minuscule scale, are doing wheat analysis, mainly doing spring wheat, just because seed to seed is a little bit shorter. But yeah, it's a very valid point that you're bringing. Maybe legumes are also an interesting system to basically tackle this, because there are a lot of studies coming from field system trials where they've used some of the legumes which are used for cover crops. So I'll definitely carry this message to Rodell for sure, because they are the ones who are handling these large scale trials. And so our Tuesday meetings every month, everybody brings their thought process or brainstorming in terms of what they're doing. And we are kind of very small piece in this large project. So I'll definitely bring this forward to them. Thank you.
Thank you.
Lindsay, I know you have another question. If you can make it short, because we're starting to run out of time for the open session.
Oh, okay. Well, sorry about that. I was just curious about the greenhouse gases as precursors to those, I guess, secondary particulate pollutants. I was curious to know whether there's any overlap there that you've noticed between the greenhouse gases being a problem for pollution from those secondary sources. That is that exacerbated in areas where you have very large dust pollution and like from the feedlots, from high erosion areas, things like that.
I don't know. So that's not, the air pollution is not that's my
Okay.
colleague's field, but I've been learning a lot about it. And I know there are areas where dust itself is an issue and can convey other toxins into humans, but I'm not positive about how that interacts with the chemistry for the greenhouse gas interactions with that particulate level.
I'm done.
Okay. Sorry to slow everybody down, but we need to thank both Jude and Harsh for being our invited speakers today and helping the committee think a lot about its statement of tasks, particularly as it refers to land management and how those practices affect the soil microbiome. And I'd like to thank everybody else who's online besides the committee and the speakers for joining us today. This committee will hold its next public meeting on Thursday, June the 22nd, and more information about this meeting is posted on our website under upcoming events. And we have many of those coming up of many different speakers, so you may enjoy those. Thanks to everyone, and especially to our speakers for joining us today. It's been very helpful.
Thank you.